Hi everyone, welcome back. Miss Calabrese here. And today we're going to be talking a little bit about the organization of cells. Some of the differences between prokaryotic and eukaryotic cells, and then a little bit about the major building blocks of cellular structure. So some of the major macromolecules that we're all made of. All right, so first off, let's talk about eukaryotes. So eukaryotic cells are ones that have organelles. So they usually have a single central nucleus and then a whole bunch of other organelles that are all going to be membrane bound. So they all have their own phospholipid bilayer membrane that surrounds all of these other organelles. And those are things like mitochondria and Golgi apparatus and lysosomes and all these other structures that are on the inside of eukaryotic cells. Eukaryotes can be single-celled, they could be multi-celled, so some eukaryotes are microbes and some are not. It just depends on how many cells are present there. So we do talk about eukaryotes in microbiology because some of them can be the cause of infectious disease. Prokaryotes, on the other hand, are much, much smaller. So prokaryotes are always going to be single-celled, so there are no multicellular prokaryotes. They're always going to be microorganisms because they're always single-celled, and those cells are much smaller than eukaryotic cells. And they also lack organelles, so they don't have those same membrane-bound internal structures that eukaryotic cells do. And then viruses are going to be very different from both prokaryotes and eukaryotes. So viruses are not cells. So we do not consider viruses to be living cellular organisms. They don't have a lot of the criteria that we set in place for what a cell actually is. So they are not cells. There is some genetic material in there, so we can see heredity and relationships between viruses genetically, but that doesn't necessarily make them alive because basically all they have is a little bit of genetic material, either DNA or RNA, but not both, and then a protein coat that surrounds that genetic material. Right, they also can't reproduce on their own. They're basically completely inactive unless they can take over another cell. So they need to essentially hijack cellular machinery in order to reproduce themselves because they don't have the capacity to do that alone. All right, so just a quick note on some of these major macromolecules. So macro just means large, so these are large molecules. These are going to be the building blocks of cells. So these are molecules that's important to understand the different classifications of them and the roles that these molecules play in the building of cells. So there are four major types of macromolecules here, and these are the things that you're built of. So you're built of carbohydrates, lipids, proteins, and nucleic acids. Those are the four major types. We consider these molecules to be polymers. And we consider them to be polymers because they're large molecules that are made of smaller repeating subunits. And those smaller repeating subunits we refer to as monomers. So the monomers think of as these small little building blocks, and then when we put a whole bunch of those small building blocks together, you get the larger polymer. So for example, if you take a lot of individual sugar molecules and you stick them together, you can get a starch or carbohydrate. If we take a whole bunch of amino acids and stick them together, we can get a protein. So the monomers are the smaller parts. If we put a whole bunch of monomers together, then we get these larger polymers. And these are the major building blocks of life. So carbohydrates are made of sugars, lipids are made of fatty acids and glycerol, proteins are made of amino acids, and then nucleic acids are made of smaller subunits called nucleotides. Okay, so back to the overall shape of cells. So let's talk about bacterial morphology real quick. So remember that bacteria are prokaryotic cells. So these are the very simple cell structure, but we find them in lots of different shapes and sizes. So here are some of the major shapes that you should be familiar with. So coccus is just a sphere-shaped bacterium. A coccobacillus is one that's not quite totally sphere-shaped. It's a little bit oblong. A bacillus is one that's rod-shaped. And then a vibrio is one that's kind of like a curved rod, so it's not a totally straight rod. And then sporillum and spirochete are ones that have this kind of like circular spiral or coiled pattern to them, right? So those are the basic bacterial shapes that we should be familiar with. All right, so the outside of bacterial cells is covered in a structure called the cell wall. So cell walls are these sort of protective walls that bacteria build around the outside of the cell. Not all cells have cell walls. So your cells, human cells, do not have cell walls. Bacterial cells, for the most part, do. Plant cells do. Fungal cells do. But those are the only ones that really have cell walls. And of each of those three that I just mentioned, so bacteria, plants, and fungi, those are all composed of different types of cell walls. So for bacteria, their cell walls are made of peptidoglycans. We talked about that word peptidoglycan in one of the previous videos. Bacteria are the only critters that have peptidoglycan in their cell walls. And that peptidoglycan is going to be made of two subcomponents, and those subcomponents are the chemicals N-acetylglucosamine and N-acetylmuramic acid, or NAG and NAM for short. And these are just chemicals that are arranged in a certain structure to provide protection on the outside of the cell. All right, so let's talk about the major differences between gram-positive and gram-negative bacteria. So we mentioned that there was a difference between these two types of bacteria before, and we mentioned that we can tell the difference by doing a particular type of stain called a gram stain. Basically, the difference breaks down to the fact that these two different types of bacteria have a slightly different cell wall structure. So a gram-positive cell wall is one that looks like this. So here we are looking at this part down here is the cell membrane. Every cell on the planet has a cell membrane, and that cell membrane is always going to be made out of phospholipids. So these are little phospholipids right here that I'm pointing at. So phospholipids form the cell membrane of every single cell. Now outside of that, here is the cell wall. So the cell wall is something that bacteria have on the outside of their cells. And this is made of peptidoglycan. And as you can see here in this gram-positive cell wall, and we have a very, very thick layer of peptidoglycan that's outside of the cell membrane, right? And that's what makes this guy a gram-positive cell, is this very thick layer of peptidoglycan. Now let's look at a gram-negative cell wall. All right, so first we're going to start off down at the bottom here and realize that the gram-negative cell has a cell membrane identical to the gram-positive cell. So that means it's still got a phospholipid bilayer, so that cell membrane is exactly the same. The next layer up is a thin layer of peptidoglycan. So right here we've got a thin layer of peptidoglycan compared to the thick layer of peptidoglycan that we had in the gram-positive. And then outside of that, we've got yet another layer, and that's called our outer membrane. So the outer membrane is something that we only find on gram-negative cells, and it's made of something called lipopolysaccharide, or LPS. Right, and that's again something that we only find in gram-negative cells. All right, so again, just another quick look at that gram-negative cell structure. So here we see the gram-positive, where we've got a cell membrane surrounded by one thick layer of peptidoglycan. Here's our gram-negative, where we've got a cell membrane, we've got a thinner layer of peptidoglycan, and then we've got an outer membrane surrounding the outside of that that's made of lipopolysaccharide. All right, so why do we care about this? Why do we care about the difference between gram-positive and gram-negative cells? And the simple answer to that is that a lot of antibiotics use these differences in cell walls to attack the bacteria. So we have particular antibiotics that work well against gram-positives, and we have particular antibiotics that work well against gram-negatives. So it's important to know which type of bacterial infection we're dealing with, so we know which type of antibiotic is going to work well. Also, the lipopolysaccharide that we find in the outer membrane of gram-negative cells, can cause some serious effects inside the body. So it acts as a toxin that your body can react to quite strongly. So if we have small levels of that lipopolysaccharide in your body, it's going to trigger an immune response, which is normal and appropriate. If we have large amounts of it, it can trigger a possibly deadly response. So that lipopolysaccharide is something that we need to be aware of. Okay. All right, so let's talk about some more external structures. So capsules and slime layers. So this is sort of like a sticky layer that we find on the outside of some cells. So if we look at some of these micrographs here, so we can see that we've got a cell. So I'll kind of highlight the inside of the cell here in yellow. So that's the inside of the cell, and then outside the cell we have this kind of thick, fuzzy layer of stuff out here. So that is going to be our capsule. So this is usually like a sticky, sugary carbohydrate material that we find on the outside of some cells. They use it for protection and they use it to stick to surfaces, so for adhesion. So if you can imagine if you're a bacterial cell, you've got this really sticky layer of goo surrounding you, you're going to be able to stick to surfaces very well. Also, that thick layer of sticky goo is going to make it difficult for white blood cells to come along and eat you, right? So that's one of the things a capsule is good for. So we refer to this as a capsule if it's just surrounding one single bacterial cell. Now, if multiple bacteria that are capsule having bacteria aggregate together and their capsules kind of fuse together into one larger structure, we refer to that as a slime layer. So a slime layer is when you've got a whole colony of bacteria and all that gooey capsule material has oozed together to form this very protective sticky layer that's surrounding the outside of the cells. Now, slime layers are really effective at protecting bacteria, and they tend to be difficult to get rid of. So once bacteria form what we refer to as a biofilm, so a biofilm is this group of bacterial cells that are protected by a slime layer and stuck very firmly to a surface. A biofilm can be very, very difficult to get rid of. So if a biofilm forms on the inside of your body, for example, antibiotics don't necessarily do a great job of treating that. In fact, the plaque that grows on your teeth that your dentist has to sort of scrape off physically because even just regular toothbrushing doesn't get rid of it, that would be an example of a biofilm. So that's when the bacteria are really adhering very tightly and protecting themselves underneath that slime layer. And it takes that physical abrasion to actually get them off. All right. Some other external appendages here. So some bacteria are going to have flagella. So flagella are like tails essentially for movement. So flagella are always for movement. So these act like little propellers that are going to help a bacteria move around in space. All right, so the main components of a prokaryotic flagellum are a filament, a hook, and a basal body. So the filament is this kind of long tail-like part of it. So we can see that here. So this is the filament. The hook is this kind of curved part here, and then the basal body is the part of the flagellum that's kind of anchored into the cell here. So the basal body is anchored into the cell. The hook is this curved part that extends beyond the cell, and then the filament is the very long tail part of the flagellum. All right, so some bacteria have just one flagellum. If they just have one, then we consider them monotrichous. So here's an example of a monotrichous bacteria, Vibrio cholerae, so the one that causes cholera is monotrichous. We can have amphitrichous bacteria. Amphitrichous means that they've got one flagellum at each end, so two. So amphi means both. Lophotrichous are ones that have a kind of like a tuft of bacteria just coming out of one or both ends. And then peritrichous means they just have flagella coming out all over the place. So they just bacteria coming out all over the surface of the cell all the way around. Right, so the point of flagella again is movement. So how do bacteria move? They're basically really simple responding to stimulus. So just kind of in the way that you also respond to stimulus to move around your environment. So if you sense something good, you move towards it. So if something smells good, coffee, cookies, whatever, you're going to move towards that because you sense a chemical that you want to be closer to. If something smells bad, you might move away from it. Um, so that's just general response to stimulus. And bacteria work the same way. So some of them respond by chemotaxis. So chemo referring to chemicals, taxis means movement. So chemotaxis means they're going to move in response to chemicals they detect in their environment. Something that they like, they'll move towards. Something that they don't like, they'll move away from. Some bacteria are also going to be responsive to the oxygen content of the air. So if we're moving towards oxygen, that'll be aerotaxis. Some of them are responsive to different magnetic fields, some are responsive to different temperatures, some are responsive to different amounts of light. So those would be all different types of movement that bacteria are capable of. Right. So in addition to flagella, some bacteria have smaller, shorter structures called pili. So pili are essentially act kind of like bridges. So a pilus is a bridge that can form between two different bacteria, and a lot of times those bridges are there to transfer DNA. When they're there to transfer DNA from one bacterium to another, we refer to it as a sex pilus. So the sex pilus is basically a bridge between two bacteria so that they can share genes with one another. Another structure that some bacteria have, again, not all, but some of them have, are fimbriae. Fimbriae are tiny little hairs that are found on the outside of bacteria that allow them to stick to surfaces. So fimbriae, if you want to visualize it, think of like Velcro, teeny tiny little hairs that let them stick to surfaces. All right. As far as internal structures for prokaryotic cells, and they don't have a lot of internal organelles or membrane-bound organelles on the inside. They do have chromosomes, though. So chromosomes are genetic structures. So this is where they have their DNA, and it's usually just a circular, double-stranded DNA, so in the form of a circle, that they keep in a central region of the cell called a nucleoid region. So there's no true nucleus, and the DNA is just usually a single circular chromosome that has all the genes for creating the next generation of bacteria. Some bacteria also can have plasmids. So plasmids are usually smaller circular rings of DNA that usually just have a few genes on them. So this isn't their entire genome. It's just a handful of genes that they sort of package separately from the main chromosome. Right. Bacteria also have ribosomes. So ribosomes are tiny organelles that we find in all cells. Whether they're prokaryotic or eukaryotic. Ribosomes' function is to make proteins. So their whole job is protein synthesis. And we've got different sizes of ribosomes depending on which cells we're in. So prokaryotic cells have ribosomes that we refer to as being 70S. Eukaryotic ribosomes are 80S. And these are measurements of size that we don't have to be too concerned with how this exactly works because you might notice that the numbers don't add up. So there's two subunits to every ribosome, a large subunit and a small subunit. In prokaryotic ribosomes, the large subunit is 50S and the small subunit is 30S. When we put the 50S and the 30S together, they add up somehow to 70S. So these are smaller ribosomes than the ones that eukaryotic cells have. So the ones that are inside your own cells are a little bit larger. Those are 80S ribosomes. All right, so some other things that we find on the inside of prokaryotic cells. Turns out that there is a cytoskeleton in there. We didn't always think that prokaryotes had cytoskeletons, but now we have evidence that they do, and those cytoskeletons kind of act in the same way as your own skeleton, right? So it's providing some internal structure to the cell. All right. We can also have storage granules. So basically little bubbles on the inside of cells that are good for storage. Usually nutrient storage we find inside those kind of granules. And then some cells, some bacterial cells also have gas vesicles, and that's where they would store gas to control their buoyancy, so to control how high or low they sit in the water column. All right, another structure here is an endospore. So I think we mentioned endospores in a previous video as well. Endospores are basically a dormant region of genetic material that some bacteria are capable of forming. So, and this is only bacteria in the genus Bacillus and in the genus Clostridium are capable of forming endospores. If a bacterium can form an endospore, that endospore can survive even when the rest of the bacteria is dead. So if the bacterial cell dies because you sprayed it with bleach or you baked it in an oven or whatever you did to kill that bacteria, the endospore can survive that. So the endospores are almost immortal, right? They're very resistant to destruction, which means that bacteria that are capable of forming endospores, spore-forming bacteria, are found virtually everywhere, because once you're capable of forming that endospore, you're basically impossible to kill. All right, so here's the process of forming an endospore called sporulation. So sporulation usually gets triggered by stress. So if the bacterial cell is experiencing stress, it's probably worried that its demise is imminent. It's going to start to form a spore. So it'll make a copy of its DNA. It'll wall off that DNA so to protect it with this very protective outer layer, so that when the cell does die, that spore remains, and that spore can hatch out later when the conditions are right and hatch into basically a brand new bacterial cell. All right, so a few quick questions to wrap up here. What's the main component of bacterial cell walls? What is the main component of bacterial cell membranes, so plasma membrane, different from a cell wall? And what's the key difference between gram-positive and gram-negative cell walls? All right, so here's your answer. So the main component of bacterial cell walls is peptidoglycan. The component of the membranes is phospholipids, and that's going to be true for bacterial cells and every cell on the planet. The cell membrane or plasma membrane is always phospholipids. The cell wall for bacteria is peptidoglycan. All right, and the main difference between a gram-positive and gram-negative cell wall, it basically has to do with the thickness of the layer of peptidoglycan. So gram-positives have a very thick layer of peptidoglycan. Gram-negatives have a thinner layer of peptidoglycan, and then they also have an outer membrane surrounding that. All right, so I hope this was helpful. Let me know if you have any questions.